Shri Rajalakshmi Prakashan published. A textbook on fundamentals of agronomy 1. Authors: Dr. D. N. Jagtap, Dr. P. B. Pawar, Dr. U. V. Mahadkar. Chapter 10th. Growth and Development, its definition, growth curve and factors affecting growth and development. Growth in living organisms may be defined as an irreversible increase in the number and size of a cell, organ or whole organism. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. Plant growth is unique because plants retain the capacity for unlimited growth throughout their life. This ability of the plants is due to the presence of meristins at certain locations in their body. The cells of such meristins have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. This form of growth, wherein new cells are always being added to the plant body by the activity of the meristem, is called the open form of growth. Root apical meristem and the shoot apical meristem are responsible for the primary growth of the plants and principally contribute to the elongation of the plants along their axis. In dicotyledonous plants and gymnosperms, the lateral meristems, vascular cambium and core cambium, appear later in life. These are the meristems that cause the increase in the girth of the organs in which they are active. This is known as secondary growth of the plant. Growth, at a cellular level, is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is, therefore, measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume and cell number. Growth in living organisms is not uniform throughout the lifespan. Growth takes place at a faster rate till the plants or animals attain maturity. Then it slows down, and at a particular time it stops. Later in life, death occurs. All these changes that occur in an organism starting from its beginning till its death may collectively be termed as development. Development is associated with morphogenesis and differentiation. Morphogenesis is the process of development of shape and structure of an organism, and differentiation is the process of change in cells, tissues or organs to carry out different functions. Stages of cellular growth. Growth of an organism is always associated with growth in size and number of cells. The growth of an organ or an organism occurs in 3 successive stages. They are: Point A. Cell division. The number of cells increases due to mitosis. Point B. Cell enlargement. The size of individual cell increases after cell division due to increase in the volume of its protoplasm. Point C. Cell differentiation. In this stage, structure of the cells changes to perform specific functions. And similar type of cells having same functions form a group, which is known as tissue. Phases of growth. The period of growth is generally divided into 3 phases, namely meristematic, elongation and maturation. The constantly dividing cells, both at the root apex and the shoot apex, represent the meristematic phase of growth. The cells in this region are rich in protoplasm, possess large conspicuous nuclei. Their cell walls are primary in nature, thin and cellulosic with abundant plasmods metal connections. The cells proximal, just next, away from the tip, to the meristematic zone represent the phase of elongation. Increased vacuolation, cell enlargement and new cell wall deposition are the characteristics of the cells in this phase. Further away from the apex, that is, More proximal to the phase of elongation lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modifications. The rate of growth of a plant or plant part is not always the same during its lifespan. Sometimes it is slow and at other times rapid. If we plot the increase in cell number, growth rate, against time, a typical S-shaped curve is obtained. This is called growth curve or sigmoid growth curve. This curve has three phases of growth. Point A. Lag phase. This is the initial phase of growth when the rate of growth is very slow. Point B. Log phase. It shows rapid growth and is maximum during the entire lifespan. Point C. Stationary phase. Here the rate of growth starts decreasing and finally it stops. The total time period during which the fastest growth of the organ or organism occurs is called grand period of growth. Growth in plants being a quantitative phenomenon can be measured in relation to time. It can be measured in terms of increase in length or growth in case of stem and root, increase in area or volume in case of leaves and fruits, increase in the number of cells in algae, yeast and bacteria. Factors affecting plant growth. Generally, plant growth is influenced by a number of factors, both external and internal. External growth factors. External factors are those factors present in the environment that affect the growth of the plants directly or indirectly. These factors are light, temperature, water and mineral nutrients. Light. There is a necessity of light for the process of photosynthesis. Besides photosynthesis, light is also essential for seed germination, growth of seedling, differentiation of various tissues and organs, and reproduction. Temperature. Some plants grow in cold climate and some in hot climate. The optimum temperature required for growth of plants ranges between 28 to 30 degrees Celsius, but it may occur in the temperature range of 4 to 45 degrees Celsius. All metabolic activities of plants are directly affected by variation of temperature. A very low temperature causes injuries to the plant due to chilling and freezing, and very high temperature stops its growth. Water. Plant absorbs water by its roots, uses it in photosynthesis and other biochemical processes, and some of it is lost through transpiration. For proper growth of plants, a particular quantity of water is required. Both deficiency and excess of water retards the growth of plants. Mineral nutrients. Mineral nutrients play important role in plant growth and development. All metabolic processes require inorganic nutrients. Plant growth is adversely affected by the deficiency of nutrients. Internal growth factors. In addition to the external factors as discussed above, there are some substances produced in the plant body itself which affects the growth of the plant. These are called plant hormones or phytohormones or growth hormones. A phytohormone is an organic substance produced in a small quantity in one part of plant body and capable of moving to other parts to influence the growth of that part. The growth of the plants can also be influenced by certain synthetic chemicals resembling plant hormones both in structure and functions. These are called growth regulators. They are not produced by plants naturally. Growth regulators are chemical substances, other than naturally produced hormones, which promote, inhibit or modify growth and development in plants. The naturally produced growth hormones are broadly grouped under 5 major classes. They are auxin, gibberellins, cytokinins, ethylene and abscisic acid. Auxin. Auxin is a growth promoter, generally produced by the growing apex of stem and root of the plants. It helps in the elongation of shoot and root tips behind apical meristem. The naturally produced auxins is indole, 3-acetic acid, IA. They are also produced by chemical synthesis, which show same physiological responses like auxin. Some of the synthetic auxin are indole, 3-butyric acid, IBA, 2,4-dichlorophenoxyacetic acid, 2,4-D, and naphthalene acetic acid, Na. The Greek word auxine means to grow. It was first isolated from human urine. An experiment was performed by Fritz went on oat seedling to see the effect of auxins. When tip of oat coleoptile, early shoot, is removed, growth stops. Then the removed tip is placed on a block of agar, gelatinous material from seaweeds, for about an hour. This agar block is then placed on the cut end of the seedling. It was observed that the growth of the seedling started again. It shows that there is something that has passed from the cut tip into the agar block, which helps to restart the growth. This was named auxin, a plant hormone. Functions of auxin. It promotes cell elongation. It suppresses the growth of lateral bud. If the tip of a plant is removed, the lateral branches begin to grow. In most of the plants, apical bud suppresses the development of lateral buds. This is called apical dominance. It delays fall of leaves. Leaf abscission, NAA, naphthalene acetic acid, is used for preventing fruit drop in apples before they are ripe. 2,4-D, 2,4-dichlorophenoxyacetic acid, acts as a dicot weedicide. Gibberellin. Gibberellin or gibberellic acid, GA, was initially isolated from a fungus Gibberella fujikurui. In plants, it is produced in embryos, roots, and young leaves, and it enhances growth. Functions of gibberellins. It helps in elongation of stems in genetically dwarf plants. By using gibberellin, the height of the dwarf plants can be increased. It breaks dormancy of seeds and buds. It induces parthenocarpy, formation of seedless fruits without fertilization, or provides stimulus received by pollination. Cytokinins. They were extracted from coconut milk. Cytokinins are synthesized in root apex, endosperm of seeds, and young fruits where cell division takes place continuously. Functions of cytokinins. They stimulate cell division, cell enlargement, and cell differentiation. They prevent aging of plant parts. They inhibit apical dominance and help in growth of lateral buds into branches. Ethylene. Ethylene is a gaseous hormone. It is found in ripening fruits, young flowers, and young leaves. Functions of ethylene. It induces ripening of fruits. It promotes senescence and abscission of leaf and flowers. In cells, it only increases the width, not the length. Abscisic acid. Abscisic acid, also known as dormin, is a naturally occurring growth inhibitor found in wide variety of plants. It is synthesized in leaves. Functions of abscisic acid. It induces dormancy of buds and seeds as opposed to gibberellin, which breaks dormancy. It promotes the senescence of leaf, that is, fall of leaves happen due to abscisic acid. It inhibits seed germination and development. It causes closing of stomata, differentiation, dedifferentiation, and redifferentiation. The cells derived from root apical and shoot apical meristins and cambium differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a tracheary element, the cells would lose their protoplasm. They also develop a very strong, elastic, lignocellulosic secondary cell walls to carry water to long distances even under extreme tension. Plants show another interesting phenomenon. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. This phenomenon is termed as dedifferentiation. For example, formation of meristins, intervascular cambium and cork cambium from fully differentiated parenchyma cells. Such meristins tissues are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific functions, that is, get redifferentiated. Growth in plants is open, that is, it can be indeterminate or determinate. Differentiation in plants is open because cells tissues arising out of the same meristem have different structures at maturity. The final structure at maturity of a cell tissue is also determined by the location of the cell within. For example, cells positioned away from root apical meristems differentiate as root cap cells, while those pushed to the periphery mature as epidermis. Development. Development is a term that includes all changes that an organism goes through during its life cycle from germination of the seed to senescence. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called plasticity, example, heterophyly in cotton and coriander. In such plants, the leaves of the juvenile plant are different in shape from those in mature plants. On the other hand, difference in shapes of leaves produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. This phenomenon of heterophily is an example of plasticity. Thus, growth, differentiation, and development are very closely related events in the life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plants, that is, both growth and differentiation, is under the control of intrinsic and extrinsic factors. The former includes both intracellular, genetic, or intercellular factors, chemicals such as plant growth regulators, while the latter includes light, temperature, water, oxygen, nutrition, etc.
We politely request, please like, share and subscribe our channel Sri Raj Lakshmi Publication.